me. Some of you may have seen back in 2007 there was an op-ed piece in the New York Times called "This Is Your Brain on Politics," where a number of people, including some of my former colleagues from UCLA, from a company called FKF Applied Research, basically scanned a bunch of swing, or the elusive swing voters of the 2008 election, and looked to see, to try to read the mind of the voter to see what's going on in the mind of these swing voters. And so they showed them videos of this is during the primary, I showed videos of all the presidential candidates, and this is some of what they found. They had a nice little slideshow. It's still in the New York Times, you can go see it there. So they give you a little emoticon to kind of interpret the brain activity, but in responses to images of Democratic candidates, men exhibited activity in the medial orbital prefrontal cortex, indicating emotional connection and positive feelings. Images of Fred Thompson led to increased activity in the prefrontal cortex, a structure associated with empathy. Subject who had an unfavorable view of John Edwards responded to pictures of him with feelings of disgust, evidenced by increased activity in the insula. Right? So they're basically using activity in particular parts of the brain to read the minds of the voters. So when this came out on a Sunday, and a bunch of friends of mine we all started emailing with each other, and it was kind of like the final straw. So we wrote an article, we wrote a letter to the editor that was published online, 15 of us, basically highlighting what the problems were with this particular approach. And we weren't the only people to notice the problem. Nature had a great editorial, and then the blogosphere kind of picked up on it. This is your brain on Hillary. And so what I want to do today is, I'll also point out that this wasn't the first time that these FKF folks had done something like this. In years past, they had done this kind of rapid imaging of Super Bowl ads. So during the halftime of the Super Bowl, they get these ads, and they stick people in the scanner right away, and then the next day in the press, they would publish the pictures of what happened when people were seeing the Doritos ads versus the Emerald Nuts ads. And clearly, people like Doritos better than Emerald Nuts because look how much more their brain turned on. And so if you look on their website, they have this great thing about how they sell neuromarketing services. They will help you design your marketing using neuroimaging, and they do it based on how the brain reacts in 9 well-known and well-mapped areas. So we can look at these areas and see whether people are feeling reward or wanting or connectedness or conflict or threat. So the question I want to ask today is kind of, can we do this? And how, if we can, how might we be able to do it? So first I'll start with how people have kind of informally tried to do it in the past, and then move on to how we might actually be able to do it more formally. So if you think about what's the kind of inferential process that we usually use in imaging, what Rick Hinson has called forward inference. We manipulate some mental process, and then this cascade of things happens, and in the end, we see an effect down here, and what we do is we infer, well, whatever brain area that we saw moving when we moved this must play some role in that mental process. This is what we call forward inference. So what I'm going to call reverse inference kind of puts that on its head, right? So basically what you do is you see activity in some area, and based on that you infer that some mental process must have been engaged. And so I'm not going to show many examples of this, but one of my favorite involves the striatum. So the striatum is, we know the striatum turns on when you get rewards. It might give you money or cocaine or show you pictures of naked members of the opposite sex. All these different types of things will turn on the striatum. So it's clearly associated with reward. So if reverse inference works, we should be able to infer that whenever we see activity in the striatum, the organism is experiencing reward. A great example of this comes from some work where people were imaging rats, doing fMRI on rats, and they were actually comparing lactating rats while being suckled by their pups compared to virgin rats given cocaine. Pretty bizarre experiment. But what they do is they basically do this, and they see that, well, lactating rats given cocaine instead of their pups showed a suppression of activity in the reward system, in the ventral striatum, basically. Thus, the data support the notion that pup stimulation is more reinforcing than cocaine. Reinforcement's a psychological concept, right? They're inferring that from activation in this particular region. And you see this throughout the human imaging literature as well. I'm not going to spend time on examples. What's the problem here? The problem here is that this is an invalid form of inference that most of us learned about when we took Logic 101, right? This is affirming the consequent. This is saying, if cognitive process X, then activation in area Z, we see activation, thus we infer that the cognitive process is turned on. That's only valid if this is an exclusive if, right? If the only thing that turns on that area is reward, then we can infer that reward is what's going on when we see it turned on. So, that's clear. But at the same time, we're not trying to build a science of deductive laws. Right? We're trying to explain how the brain works. And it may well be that there's some information to be gotten from this, despite the fact that it's logically invalid. And so the question, people sometimes refer to this as, abductive reasoning to the best explanation, right? So we wanted to ask the question, can you tell how useful a reverse inference might be? Is it useless or not? And we started by thinking about it probabilistically, right? So if you think about, what do we know? We know, let's assume that I know that if I, turn on some particular task, then that causes this particular cognitive process to be engaged. So whenever I make you do the rhyming task, it causes phonological encoding to be turned on, for example. Then we can do an experiment and find what's the likelihood of, or we can look at a bunch of experiments or a bunch of subjects and find what's the likelihood of activation in a particular region, given that a particular cognitive process is engaged, right? What's the conditional probability of that? What we want to know for reverse inference is the flip side of that, right? We want to know the likelihood of the cognitive process being turned on given the activation in that area. And so we can use Bayes' theorem to get that, right? And you can sort of think of Bayes' theorem, I'm going to use it in sort of reframing it this way. Think about, the conditional probability over the marginal as sort of being the selectivity. How much more likely are you to get activation for that particular cognitive process than you would for sort of all cognitive processes? How selective is activation in that region? So we can ask then, is there some way to tell how selective activation is? And there's no really good way unless we could sort of have data across all possible tasks. But we can kind of make a first stab at it by using databases that contain data from lots of different imaging studies. So we worked with the brainmap.org database, which when we did it contained data from about 1,300 papers, a total of about 46,000 activation locations. So what this database stores is like the table that Bill showed you yesterday with a bunch of little activation locations. For a paper, it will store all of those and then reconstruct 3D activation patterns from those locations. Okay, so we worked with Angie Laird at UT San Antonio to try to just test a particular reverse inference. So one particular reverse inference that you sometimes see is Broca's area was turned on here. Thus, the person must have been engaged in some kind of language process, right? And you see this fairly regularly. So what we did was we asked, well, how well can you actually tell? If we look across all the brain map studies, how much more likely is activation in that area when language is turned on compared to when it's not? And we have to just rely on the fact that each of the different comparisons that's in the database is coded as to whether it's, in theory, isolating some language process or not. That's not perfect, but it should give us something, right? So when we do that, we see that most of the studies don't activate Broca's area, but a good number do. And more do for non-language studies than language studies, but you can see that the relative likelihood is higher when it's a language study than a non-language study. And if you work this through, and we'll just assume a prior of, we have to assume a prior to get a number out of this. Let's just assume a prior that language is engaged of half, so there's a coin flip. If we then, use the data here to get a posterior, the conditional probability that language is turned on, given that they're doing a particular task, which we had no prior about, and that Broca's area is turned on, it's a little bit higher, it's 0.69, right? So there's some information in there that's telling you that it is more likely that language is engaged. It's relatively weak, so the Bayes factor is 2.3, and Bayesians generally think that anything below 4 is pretty weak. So there's some information there, but it's not particularly strong, at least for this particular thing that we looked at. Now it turns out that if you reduce the size of the region, right? So if you had a really big region, obviously lots of things can turn that on. As the region gets smaller, selectivity goes up. So if we move down to a 4 millimeter region of interest, centered around, we just took a point from a study that had done a bunch of different imaging comparisons and kind of isolated what we think is this area that's doing phonological processing. If you go for the really tightly for that little area, it gets more selective, so you get a base factor of about 4.5. Still not great, but at least it's it's not quite as weak. But rarely are people saying, oh, I'm going to assume that language is engaged because I had activity in this voxel, not in the one next to it, right? It's generally much more sort of loose than that. It's really centered around kind of broader anatomical regions. So generally, the size of the ROIs that people are doing reverse inference to are usually bigger, probably 40 millimeters or so. So what I argued in this paper published a few years ago, which kind of presents all these data, is that informal reverse inference of this kind really provides pretty weak evidence. But if you look, I used to refer to this as the NeuroImage discussion section syndrome. NeuroImage is this journal, and if you pick up any paper from that journal, almost any paper, you look and there would be sections titled anterior cingulate cortex, and it would go through and say, what do we think the anterior cingulate cortex activation means? Oh, it means conflict or blah, blah, blah. And often that's because a reviewer would say, oh, you have activation in the anterior cingulate cortex. What does that mean? Right? And so it kind of drives, again, it's, I think there's some interesting things about, what reviewers ask for, which ends up kind of mucking things up, similar to what Chris talked about. There's interesting reasons to ask why activation might be so weakly selected. There's a lot of reasons that it could be. It could be that function really is distributed, right? It could be that it's not distributed, but we're at the wrong resolution to see it, or it could be that the things that we're trying to map don't exist or are incorrectly chopped up. That is, that our ontology is wrong. And that's an issue I'm going to come back to in a little bit. Okay, so but first I want to ask, can we actually do better, right? So we see that this kind of informal reverse inference is not particularly good, right? Can we do better? And an obvious place to look is the set of methods that have been developed in the last 20 years or so in machine learning, or whatever you want to call it, that give you statistical methods for predicting things, right? So the idea is, and you're going to hear a lot more about this later, you have an fMRI data set, you train your statistical tool on part of the data set and then test on the other part, right? So here you can imagine asking exactly the same kind of questions that you want to ask with reverse inference, but doing it in a formal way by taking part of your data, training it to detect conflict or fear or whatever, and then testing on your other data to see if it's actually there. I don't know if you're going to show examples of these different things, but I'll just sort of mention it. This has become a huge thing in the literature. Applying these techniques to fMRI data, and you see that certainly for visual stimuli, this is the sort of first paper that really made a splash in this stuff. You can get highly correct classification of what a person's looking at. John Dylan Haynes and his colleagues have done stuff looking at more, sort of higher level cognitive processes. So this is an example of a study where what he did was he gave people a cue, and he had them decide when they got the cue. So he said, you're going to see some numbers in a moment. I want you to decide whether you're going to add or subtract those numbers. So the cue comes up, you decide whether to add or subtract. Then the numbers come up and you add or subtract them. And then you get a cue to ask you what the answer was. And that's how we can tell whether you added or subtracted them. But what he wants to do then is look at brain activity that's evoked out here and see if he can predict whether you're going to add or subtract. Okay, and it turns out that he did this using the searchlight technique where you sort of move around the brain and see which regions give you information about that decision. And you see that, for example, here in the cingulate cortex, you get some regions that can give you about 70% accuracy in predicting whether the person is going to add or subtract. Now, this led the press to go crazy. Brain scan sees hidden thoughts, read minds, mind reading machine knows what you see. And one of the points that I think should come out of this is the need for more care in thinking about how we present these things to the world. And as a canonical example, I want to show you some video of one of our colleagues, Marcel Just. Let me see. Turn it up so you can hear him.
Do you think one day, who knows how far into the future, there'll be a machine that'll be able to read very complex thought, like, I hate so-and-so, or, you know, I love the ballet because.
Definitely. And not in 20 years. I think in 3-5 years. Ample
In 3 years.
Well, 5.
So clearly, you know, some people are very bullish on the power of these things, not just to tell what kind of contents we have, but actually to read out, not just tell what, you know, what kind of things we're looking at, but to read out complex, you know, contents of thought. But this makes a particular claim. You missed the video of Marcel.
Oh, please replay it. Come on. Come on. It's my daughter. I had to take it.
It's going to give us all a copy later.
So, you know, one of the claims that when you ask people, when you think about what people on the street mean by, or think of when they talk about mind reading, it's generally looking at the brain of someone you've never seen before, right? All of the stuff that, almost everything that's been done in literature has been scanning a person, taking part of their data and training the classifier, and then taking the other part of their data and testing the classifier, right? So it's within subject prediction. And so, but you know, people on the street, when you ask them about fMRI mind reading, they think about you like, you know, reading the mind of somebody to decode their intentions when you've never met them before. And so we wanted to ask the question, can you classify mental contents in people you've never seen before? So can you generalize across people? And so we did this in kind of a funny way where basically I just took a bunch of studies we had done in my lab using very different tasks. One of the benefits of being all over the place is you have data on people with the same scanner doing a bunch of different things. So we had 130 subjects total, each of whom had been in one of 8 studies doing different types of tasks. And the tasks are really all over the place, from you know, decision making and a sort of risky decision making task, classification learning, rhyming judgments, auditory working memory, gambling decisions, living, non-living decisions, reading words aloud, and response inhibition. They varied between block and event-related design, all collected on the same scanner, all analyzed the same way, but you know, relatively different in what the subjects were doing. For each of those tasks, we processed all the data the same way, and for each task we got a statistical map for that task versus sort of a resting, like a visual fixation baseline. So for each subject, we just compared doing the task versus baseline, a really gross comparison, right? So if it doesn't work here, it's not going to work anywhere. So for each subject, you get that Z statistic map, so you have a Z at every voxel for the task versus rest. And then we do some various types of feature selection and then some various types of classification, and I'll talk more about those in a second. But the idea is, can we predict which of those 8 studies the person had been in when the classifier has never seen their brain before? It's only seen the brain of we did a leave one out cross-validation, so it's only seen the brain of the 129 other people. Now we classify number 130, we loop through and do that for everyone. And here is what we find. If we take voxels where every subject has data, because remember we have this initial processing and some subjects are going to have missing data from certain places due to dropout and things like that. So let's just take the voxels where every subject has data. We can get this generalization accuracy of about 80%. Chance here, if you just randomize the labels and do that over and over, you see that anything above 18.5% is significant at p less than 0.05. So we're way beyond that. If we only take the voxels, we can also take these Z statistic maps across all the 130 subjects and find what voxels were activated on average. So what voxels had a Z greater than 0 significantly. We call those the positively activated voxels, and that's just activated across all these tasks. And we can also take the negatively activated voxels, the ones that are less than 0. And those fit with there's a set of areas that are always deactivated when you do any kind of difficult cognitive task. That's the red, yellow stuff here is stuff that was activated across all these tasks. The blue stuff is stuff that was deactivated across all the tasks. If we look in the positively activated voxels, you see that we can still do pretty well. We can do 74.5%. If we look at the deactivated voxels, you can still do above chance, but not quite as well. There's fewer of those, so it's hard to know how much of that is just driven by the amount of data. But it does suggest that all of those voxels are going to use some kind of information. This is a confusion matrix showing the actual tasks that they were doing compared to the tasks that the classifier thought that they were doing. And you see that there's one particular task where it confuses a lot between these two. And it kind of makes sense that these are both tasks where you're just basically making judgments without any feedback. It's a gamble decision task and a classification decision task. So in some ways they're fairly similar. The rest of them, the rest of the tasks it does fairly well on. This just came out in Psychological Science a couple weeks ago. Another thing we can do is look around the brain and see where, how well can you do just in any little local area. So this is, again, using the searchlight method. So you basically move a little sphere around and then, say, within this little sphere with a radius of 4 voxels, how well can I classify? And you assign that accuracy to the center sphere, and then you move to the next, and the next. The best you can do in any little area was about 44%. So you're still above chance, but nowhere near that 80%. So it suggests that there's some local information, but you also have to integrate information across regions to really get to get up to the kind of ability that you can do. And similarly, if you break it up into anatomical regions of interest, the best anatomical regions are at around 50%, and those are primarily motor cortex and visual cortex. But there are some, like anterior cingulate can get you 40-something%. So it's not just basic regions. There's also higher order regions that seem to be providing some kind of predictability. So we can accurately predict across people what they were doing, right? Without ever having seen their brain before. So it's these kind of data that lead me to often kind of poo-poo issues that are brought up about individual variability and small differences in, these are, I should mention, these are spatially smooth data, I think with a 6mm kernel. So clearly, despite all of that individual variability, we can predict fairly well. I'll mention that we did some comparisons where we had some of these people doing multiple of the tasks. So we could train the classifier on that person doing one of the tasks and then include them at test doing a different task. And what we saw was, for the most part, the classifier was more sensitive to the task than it was to the person. It differed across tasks. One of the tasks, it was really good at classifying the task. The other one, the accuracy was less and it got more pulled towards the person. But it suggests that even there, you can it can pull apart the differences between tasks within an individual. Okay, now I've talked about kind of tasks, right? And we're cognitive psychologists, that's what we do, we come up with tasks. But in the end, we're only doing that as a conduit to get to the mental processes that we think live below those things, right? And in the end, what we'd like to predict is the mental processes, like fear or conflict. We don't want to predict what tasks you're doing, because in the world, people don't do tasks. But what that requires is first that we know what the things are, right? What is the ontology of the mental processes, and how do they relate to these tasks? So, for those of you who are familiar with this term, and it has different usages in philosophy and more recently in sort of informatics, and I'm going to sort of stick with the informatics framing of it. It's basically a formal specification of what exists in some domains. So, for example, Wikipedia shows this little ontology for vehicles. Cars and trucks are kind of vehicles, two-wheel drive is kind of car, and so on. And in molecular systems biology, there's been a huge move towards using ontologies to help organize data. So for example, if we want to ask the question, what biological function is some gene associated with? What people generally do now is they don't write a paper and say, oh, I think, blah, 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 it does this. They say, I think it does this. And then they tag it with a tag from this thing called the gene ontology, which basically provides descriptions of all known gene products and functions. So the idea is we don't just want to write what we're talking about. We want to formally specify it. And what that does is, one, let us all sort of agree on what we're talking about, but also couch it in the context of larger things. So for example, if we're talking about immune response, we know that's a kind of immune system process and that's a biological process and so on. So it lets us make some further inferences from that particular process. And there's all sorts of incredible tools now that one can use based on gene ontology to try to get at, given some particular genetic data set, to get at what its function is. But psychologists haven't, it's interesting, since the faculty psychologists haven't really focused on structure, right? We focused on function. If you look at cognitive psychology books, they talk a lot about functions, but they don't talk a lot about, what's the larger structure of the mind, right? How are different functions related to one another? And there's nowhere you can look, there's no book you can open up. So for example, there was a guy at UCLA named Paul Boyer who's famous for writing this set of books called The Enzymes, which is like a 10-volume set that lays out, what are all of the enzymes that are known about. There's no similar place where we can go look for the mental processes or the mental functions, right? Nobody has cataloged what we think they are, other than doing it sort of informally. But I want to argue that if we're going to get anywhere, and I think Bill, because I think this sort of, is similar to an argument that he made in his new phrenology book, that if we want to take this seriously, we really have to think more formally about what is it that we're actually mapping onto the brain. We can come up with kind of informal ways of thinking about it, but if we really want to get anywhere, I think we have to formalize it. So we've been trying to do this in this project that is crazy, I think, but somebody was crazy enough to give us money to do it, called the Cognitive Atlas. And it's almost ready for people out in the world to see and play with. So within the next month, if you go to cognitiveatlas.org, it will be available for people to log in and actually start doing some stuff. And the idea behind the Cognitive Atlas is really, we think that cognition sits, this actually task should be kind of out to the side, not in the middle here, but the idea is that, I was hanging out with psychiatrist type people, and they're really interested in, how do cognitive processes fit in how we think about, psychiatric syndromes and how those relate to the brain. And so the idea is that, there's this set of cognitive functions that relate things like schizophrenia to things like brain systems and signaling pathways. So the Cognitive Atlas aims to basically provide a formal description of what all the things are at that level of cognition and how they relate to particular tasks. And this will basically be done, in a Wikipedia type way, which means that we're not going to be doing it ourselves. We're going to rely on the community to help do it. And I'm happy to show more about sort of what that thing's all about. But the goal will be in the end to have a knowledge base that provides the basis to annotate any particular data set, right? You have a comparison between two tasks or two conditions, and this would provide what the community thinks, or what different people within the community think that comparison maps onto mentally. So we don't have that yet. We don't have that knowledge base, so we can't really do any analyses with it, but we can at least do kind of a proof of concept to suggest that if we had that, how might it work? And so what we did in this paper was we took data from those same 8 tasks, and we coded the tasks using a kind of a coarse ontology of mental processes. So basically we say, for each of the tasks, which of these mental processes is engaged. Then we reduce the dimensionality of the data and try to map the data on, through the tasks onto those mental processes. So let me show you how we did that. So first, I just sat down and we took this set of very coarse labels from the brain map database. They have this thing called the behavioral domains classification. And we basically went through for each task and said, okay, does this task involve language? The ones that have white are the ones where I thought that task involved that particular process. So again, this is wrong in a number of ways, but it's useful as a starting point. Then we take the data, and so that we started with the same data that I had done the classifier with. The data had about 200,000 voxels at the resolution we were using. And we wanted to do a dimensionality reduction, but we wanted to, we could have done something like ICA or BCA, but we wanted to actually do something that was sensitive to the differences between the tasks. So we actually used a neural net where we trained the neural net, given some particular input pattern, to tell us which of the 8 tasks was being done. And then we couldn't give it 200,000 voxels as input just because of computational reasons. So what we did was we took the 1% of voxels that just had the highest entropy across subjects without regard to what tasks they were doing, and put those in. So basically, it's we took the highest 1% of the data in terms of informativeness. And what we then do is we train that up, and we varied the number of hidden units, and it turned out that basically, if you went above 6, you didn't really do any better. And so we took 6 as our sort of standard here. With 6 hidden units, you can get about 75% accuracy. So that suggests that, I don't have a picture of where the voxels came from, but they were kind of scattered all over the place. So, so we can do fairly well even with that very sparse representation of the data, which isn't surprising given that the data were smoothed. So the first thing we can do is just look at sort of clustering, and so this is the data. So we take the data from the hidden units for each subject. We train the network, then for each subject give it their 2,000 voxels, and we take out the values of the 6 hidden units for each subject, and that's what you see across here. Each column is a subject, and each row is a hidden unit, and the grayscale is just positive or negative values of those hidden units. And then we clustered across those, and this is a color-coded thing that, so each different color reflects a different task. And so what you see is that you get some clustering of tasks, as you would hope, given that the classifier actually works. But what's interesting is it actually kind of puts things together in more reasonable ways, too. So for example, reading and rhyming and semantic judgments all go together here. Working memory and inhibition, which are the 2 tasks that involve auditory stimuli, are over here. So there's some kind of structure that seems to be coming out of it. But it's still a little bit messy. So then the question is, how can we take this and move towards, actually figuring out something about what these dimensions mean, right? Because presumably those dimensions are kind of the bases on which these 8 different tasks differ. And the question is, what mental processes might be mapping onto? So what we do is, we take this concept by task matrix, then what we do is for each of the 6 dimensions, we just average across subjects to get this what we call task by dimension matrix. And then we project those to get this concept by dimension matrix. So for each of the concepts, what's its mapping on each of those 6 neural dimensions? And so here's what we find. So it's kind of a complex graph, so let me unpack it. So at the bottom, what you see is a mapping of it's basically the input to hidden unit weights for the voxels, for all the voxels to this particular hidden unit. So this is for one of the hidden units, or one of the dimensions, right? And then we smooth those out because the voxels are kind of all over the place. So it's basically it's the weighting of each voxel for that particular dimension, spatially smoothed. That's what's down here. Up here is basically a tag cloud representation of each of those different concepts. So the bigger the concept label here, the more strongly it was weighted by that particular dimension. And clearly there's a lot of reasons why this is. I don't want to suggest this is telling us anything new about the brain. I just want to show it to you and try to convince you that it tells us something sensible, such that if we actually had what we needed to do it for real, big data sets and a big knowledge base, then it might actually tell us something new and interesting about mind and brain. So for example, here audition is the thing that pops out. These other things that are sort of related to audition and the tasks that we had in our database pop out, and you get right auditory cortex, and then this thing in the medial prefrontal cortex. Here's one for decision making. Decision making is the big thing that pops out, and you get basal ganglia, some medial prefrontal stuff. You get another fairly similar pattern for a different one of the dimensions. So the dimensions ended up being fairly similar. I'm sorry, you had a question?
Just a clarification. So the selection of the 2,000 was done without regards to location or there being clustered or anything
Exactly.
like that?
We just took the 130 subjects and computed entropy across them at that voxel, and then took the 1% highest without regard to location or to task.
And even then, the ones that get connected to that hidden unit happen to be this nicely localized.
So this is another of the dimensions that comes out, and what's interesting here is that this really maps pretty closely to the response and emission network that we've talked about in a number of recent papers. You get basal ganglia, you get right inferior prefrontal cortex. Again, it's perhaps not that surprising because there's a small number of tasks, and one of them is the task that we've used to make that argument. But it does suggest that it can help you get out interesting answers that, if you had a large enough data set, could start to tell you something about the structure of cognitive processes that map onto brain systems. So now we're no longer mapping tasks to brain systems, we're mapping cognitive processes to brain systems. And so the hope is, I have this fundamental conviction that a lot of the concepts that we have about mental function are simply wrong. We're just chopping up the mind in the wrong way. So I view this as a way to help us figure out how the brain chops up the mind, and by doing that, helping us kind of reframe how we conceptualize mental processes. So for example, if you saw that, let's say, working memory and task switching were something that you had, data from a bunch of different studies on, but you saw that they always popped up in the same, in association with the same neural dimensions. That might suggest to you that those aren't really different concepts. Right? Likewise, if you had things that were always discriminated, or if you had a particular concept that was popping up in multiple dimensions regularly, it might suggest that you need to think about breaking those up. So the hope is that this can give us a way to start thinking about reframing our understanding of mental processes using what the brain tells us. And one thing we need for this is big databases, and we just don't have them yet. fMRI researchers have been terrible about data sharing. It's just failed so far, basically. But that's, I think that's a travesty, given how much money goes into fMRI data, that it's not all shared. But it isn't. And but people are now working on finding ways to do it. It's a difficult problem because the data sets are so big, the metadata is so complex, and up to now we haven't had frameworks to characterize the metadata, but that's coming around. And so I think that within, say, 10 years, maybe we'll have databases that have enough data in them, and we'll have our knowledge base hopefully sort of fleshed out well enough that we can actually ask these kind of questions. Okay, so we can't read minds yet, but we're moving in that direction, depending on what you mean by mind reading. I still continue to maintain that the kind of reverse inference that's still used heavily in the literature, though people have become more sensitive to it, should be treated with caution. The classifier stuff is, I think offers us some really cool ways to think about how we can answer the kind of questions that people want to answer with reverse inference. And hopefully that'll provide us a way not just to classify, what kind of stimuli people are looking at and what kind of tasks they're doing, but to really rethink how mental processes are put together. So I just want to thank particularly Steve Hansen and Yari Palchinko, who worked on the classifier stuff with me. Thanks.
Thanks very much.
Okay, we have some time for questions before. Yeah, George.
Well, I was just wondering, it seems like it would be useful to characterize the amount of information you're getting in an informational measure like bits. And it seems like you're never getting more than 2 at most 3 bits.
Out of what?
Out of the classification paradigms.
Yeah, if we had, so what we need is bigger data sets, right? We've started doing some stuff like this with the brain map data set, with the brain map database, where you can, for example, you can have, all those ontological elements that I showed. You have multiple studies that maps onto each one of those. There you can start getting up to 40 or so alternatives and trying to test it, with a little more information. And we've done pairwise comparisons there, and that works. We haven't done it in kind of one versus all.
The other thing I was going to say is that a very similar enterprise has been going on in EEG for people with ALS, so that they can communicate by their brain activity.
Right.
And you might look into, excuse me,
the BCI stuff, I think, the brain-computer interface stuff is clearly, some of the questions that they're trying to ask are very highly relevant. For the most part, they've been more focused on things like moving robotic arms or typing or things like that. But I think you're right that in the end, they're trying to solve the same problem that we are.
People actually have gotten reasonably good.
Mhm.
I think at least as good as with fMRI.
Right.
Even though the signal in some sense is inherently much weaker.
So I guess one point is that, I think it's fantastic and it gives one a lot of descriptive understanding and a lot of dimensionality reduction.
Right.
But the fundamental, almost epistemological problem remains that one has no means of simply by doing this analysis of making an inference about where the causality lies. So the fact that you can get different patterns that accurately classify different mental states does not in itself license the conclusion that those patterns in the brain are what cause those mental
Sure.
states.
Yeah. No, I think that, but that's just a fundamental problem with imaging,
Yeah,
right?
no, I agree.
Yeah.
I'm not trying to be critical. I'm
Yeah.
just trying to.
Yeah, but so I guess the way that I view it is, what in the end, this is about hypothesis generation, right? This is about driving us to do new experiments that we wouldn't have thought of, or in the end, I don't know if you saw this thing. There was a thing in Science, I think last year, about the automation of science, where they built this thing that basically sort of figured out what the right experiment is to do next, and so on and
I
so.
wanted to get one of those.
Yeah. Clark.
Well, maybe it's the last one before.
You had a slide where you made some comment about individual points not giving you good predictions in your, what was the domain task, binary decisions. And the argument was that therefore you needed a whole lot of points. But I wonder whether you explored how many points you actually need. That is to say, do you really need a whole lot, or would 5, 8, 10 do?
Right. So, for example, could we get up to 80% just using visual cortex and auditory cortex?
For example.
And we haven't done that. Yeah. I think that's a really interesting thing to explore, to figure out what it becomes a combinatorial mess to figure out how to do it right. But yeah, we haven't done that.
Okay, shall we?